Our next speaker is going to be Enakshi Singh. She's a product manager for the SAP HANA Platform for healthcare. She's going to tell us about how this platform enabled new discoveries in genomic research.
Thanks, Liz. So before I start, I kind of want to do a quick poll. How many people have heard of SAP? So about half. How many people have heard that SAP is doing something in genomics? Like 5 people. So my talk's going to be focused a little bit more on the research side of genomics, but as we all know, we don't know enough about the genome, so this is equally as important. So just a quick background for those of you who haven't heard of SAP. SAP was founded in 1972. Its headquarters is in Waldorf, Germany, and we're a market leader in the technology software for business management. What that means is basically SAP creates software to help small, mid-sized, and large companies run better by creating software for things like sales, management, HR, and finance. We have over 61,000 employees and over 12 million users, so it's safe to say we're a pretty large company. About 5 years ago, SAP released SAP HANA, which is an in-memory columnar store database. Without getting too technical, the point of, SAP HANA has basically revolutionized SAP's core business. With HANA, we can speed up the way analytics is done with traditional software systems. So I'm not going to really go into this detail. I'm not a computer scientist, so I probably couldn't, even if I wanted to. But to highlight a couple of the features of SAP HANA that we've seen that's helped with our genomics and clinical work. Some of them worth highlighting, I guess, are the text retrieval and extraction engine. So HANA actually originally started as a text retrieval and extraction engine, meaning you can search unstructured data like doctor's notes. And that's proved useful for us in an application that I'll talk about a little bit later near the end. And with HANA, we can compress the data, and so we can fit all of the data in memory, and that makes it quicker to be able to analyze all of this data. And in this context, we're talking about genomic data. So why does everybody care here? So our vision with the SAP HANA platform for healthcare is to integrate all relevant data that might be important for patient treatment. So we've kind of siloed data into 3 different umbrellas. The first is the biological data, which includes genomic data, but also all other omic data like proteomic data, metabolomic data, can all be important, and integrate that with clinical data from electronic medical records, and then finally lifestyle data from things like wearables, the Fitbits, and the Jawbones of the world. And so what we envision at SAP is HANA being the enabler to integrate all of this data, and the challenge that we've given ourselves is, can we enable the integration and analysis of all this data while a patient is sitting there with the doctor? So a couple years ago, about a year and a half ago actually, we started working with Dr. Carlos Bustamante at the Stanford School of Medicine. Carlos is a statistician by trade, but his lab focuses on population-level genomics. So what that means is he's looking at large-scale genomic data sets and trying to find patterns of variation within and between species, and ultimately he wants to contribute to a better knowledge of how genomics contributes, or genomic variation contributes to health and disease. So we started working with Carlos, and we took a look at the genomic processing pipeline, and you guys are probably all familiar with it. You sequence a genome, then you need to align it to the reference, call the variants, and then finally need to annotate and analyze it. So the reason that the last 3 steps are in red is that we think that, or we thought when we started working with Carlos that we can help have SAP HANA contribute to these steps. So the goals of the collaboration were to provide SAP HANA as the platform to do the real-time analytics of large-scale genetic variants, and to provide the basis to integrate with annotation data, and then to finally be able to use whichever processing pipeline for alignment and variant calling you want in the SAP HANA Genomic Analyzer application that I'll have in a second. So most of you are probably familiar with the variant call format. This is the emerging standard for storing genomic variant data. When you show this data format to the computer scientists at SAP, they are honestly appalled. So there are a variety of issues with this data format. So it's highly dynamic. You can add as many columns as you want as long as you define the metadata at the top. So all of the pound lines are the metadata, which then define the columns that succeed it. And the format fields are typically the most important fields, which give you the genotype information of your sample. And that's all the way hidden over there. So think about trying to figure out how many patients have this particular genotype with this kind of data set. You're essentially writing a manual script and waiting a really long time for it to run because the data is not accessible. So one of the very first things that we did was we took this VCF format and mapped it to a data model in SAP HANA. So SAP HANA has many things, as I kind of briefly showed, but it's also a relational database at the core. And so by mapping this data model, which was not an easy feat because of the dynamic nature of it, but somehow our colleagues figured out how to do that, naturally we need to benchmark it. So what we did was, working with Carlos and Dr. Tulbute, who you guys will probably see tomorrow, we took the 1,000 Genomes project dataset, the pre-phase 1 release, which includes the genomic variant data of 629 individuals, and we took a disease database that's been curated manually by Stanford. It's called VariNet, and it basically includes a variety of GWAS studies that show the associations between certain SNPs and disease. So there are about over 500,000 SNPs in that database and about 2,000 diseases. So what we did was we took this 1,000 genomes data set, loaded it in HANA, and then also loaded this disease database in HANA and joined it together, a simple database join, and then segmented the results by population. So there are 4 main populations in the 1,000 Genomes Project data set. I hope I didn't lose you. So just to kind of put it in a different context, we wanted to know how the genetic variants are expressed across different populations. And we did this for about 8 different diseases that Stanford told us to look at, and we found something interesting with type 2 diabetes. So type 2 diabetes genetic risk has a huge disparity based on the ethnicity that you are. So it appears that people from the Americas have a significantly higher genetic risk than people of East Asian descent. So why is this important? This is important for many reasons. One is that the reference data that we're using is highly biased towards European populations. And so if somebody like me of Southeast Asian descent is told that I have a slightly higher risk for type 2 diabetes, that's based on data from Europeans, which may not be accurate for me. And so to kind of also explain how important this was for Stanford and Tulbute actually had published this exact same finding with a much smaller data set. So with this, we looked at 125 genetic variants associated with type 2 diabetes across all 629 individuals, and did this in under a minute per chromosome with SAP HANA. This paper that was previously published with the same findings looked at 12 genetic variants in 49 people. A much smaller data set because the technology today is pretty crude in analyzing genomes. And so the fact that we were able to corroborate those findings with a much larger data set further shows that there is a huge disparity with this disease, and I mean, who knows what we'll find if we start looking at other diseases across different ethnicities. So since our learnings of this finding, we basically adapted the data model and simplified it, and since then we've loaded the phase 2 release of the 1,000 Genomes Project data. That includes the genomic variant information of 2,534 individuals. That is 1.2 terabytes in HANA and translates to 22.4 billion records in HANA. So for those of you that are not database people, to put that in context, last week we were at the largest SAP user conference, the annual user conference called Sapphire. Our CEO gave a demo on the simplified financials application on HANA, and that demo had dummy data of all of the SAP financial information, which translated to 380 million records. So all of SAP's financials, which remember we have over 100,000 customers, is 380 million records. About 25,000 genomes is 22 billion records. So there's a lot of data. And with our data model, we can flexibly and interactively query across all chromosomes, across all data, and we can easily integrate with external annotation data sets by doing simple database joins. So with that, here is a screenshot of our application. It's pretty simplistic. Essentially, it provides the user, which at this point is a genomic researcher, to be able to analyze genomic variants in whichever way they want. So a user can go into this application and pick an analysis that's already been done, or they can create their own template. So I can't show the demo here, but if someone is interested in seeing it, please come by our booth and we'll be happy to show you live. So the way that this application works is it's simple SQL queries. Once you understand the data model, you can just design your query yourself. We understand that most people are biologists and not SQL query designers. And so what we've done is we provide the ability to reuse query templates that have been written by other researchers. Ultimately, the types of queries that you're doing across genomes are pretty similar with a variety of use cases. And yeah, so like I said, if you guys want to see this live, we can show you at our booth. And we're basically we're able to query all 22.4 billion records in seconds to find a simple association. So the screenshot at the bottom is looking at a single SNP associated with the alcohol flush reaction, which I actually took from my 23andMe, and we just queried that across all individuals, and we can see that there's about 380 people that have this G genotype there. Right now, we're in this beta testing phase of the Genomic Analyzer application. What that means is we're looking for people who are interested in analyzing genome data, and what we'll do is we'll provide you with access to our system, and we'll preload the 1,000 genomes phase 1 data set, which contains 1,092 individuals. We can't share the 2534 individuals yet because it's not yet released officially. And the idea is that you would work closely with our team to develop queries and even integrate the 1,000 genomes data set with external data sets like cell expression data, clinical data, and things like that. So if that's something that's of interest to any of you, please come find us after. So moving forward, we're going to continue our collaboration with Carlos, and we're also excited that a couple other professors at Stanford will be collaborating with us. So Ewan Ashley was the guy who led the team in 2010 to the first clinical interpretation of the whole genome. He's now director of the Genome Clinical Services in Stanford, and also the Stanford Center for Inherited Cardiovascular Disease. And Atul Butte, he's doing a keynote tomorrow. He is very interested in looking at I think his keynote is called taking 3 trillion data points and trying to find diagnostics and therapeutics out of that. So we're going to work with these professors in their lab to ultimately enable new findings to understand how human genetic variation contributes to health and disease. And we'll focus on cardiovascular disease because Dr. Butte actually is interested in that. And then we'll also enable large cohort studies and try to find, again, associations between genetic disease. In addition to that, we recently partnered with the Global Alliance for Genomics and Health. So some of you may have heard of that. It's an international alliance recently formed, and the goal of this alliance is to enable the sharing and ethics of genomic data. And so we're very excited to be a part of this global alliance as well. And in this global alliance, there are 4 working groups. Naturally, we're going to gravitate towards the data working group and hopefully be able to contribute to the analytics part with SAP HANA. And then lastly, I kind of wanted to mention another application that was built out of SAP Germany with the National Cancer Center for Tumor Diseases. So the NCT is both a research center, but also a cancer treatment hospital. And what our colleagues in Germany did was they took SAP HANA and integrated the various structured and unstructured data sets at the NCT, which included tumor documentation, medical records, and clinical trials for over 150,000 patients, including over 3 million interactions, and basically created this front-end UI to be able to do ad hoc filtering and analysis. So the screenshot that you're looking at is looking at the number of patients that have been diagnosed at the NCT with non-small cell lung cancer and their association with certain biomarkers. So it looks like non-small cell lung cancer has a high association with EGFR. So this type of analysis couldn't be done at NCT before because the biomarker-specific information was sitting in an unstructured data set in a doctor's letter. So what we did with SAP HANA was we were able to extract that unstructured information and put it in a structured way by just creating a set of rules to take that biomarker-specific information from the doctor's letters, put it in a structured way, and that way we can integrate it with the rest of the data. So one of our next big goals is to integrate the functionality of the Genomic Analyzer that I just talked about with this Patient Data Explorer tool. So picture being able to look at all of the electronic medical records at a hospital in conjunction with all the genomic data and be able to find associations historically to ultimately get to that evidence-based medicine rather than practice-based medicine. So with that, thanks, and if there are any questions, happy to answer them.
Any questions? I have a question. You mentioned towards the end ingesting clinical data. Of course, there's lots of problems. If you think VCF is bad, clinical data is a million times worse in terms of its lack of order, structure. And would you see yourself partnering with someone around ontologization of the data, or?
Yes and no. So we're very aware that every EMR has a different structure and there's a lot of overlap. So the way that we probably go about doing it is to find a specific partner and create an adapter for that particular electronic medical record. And as we continue to do that, we'll have a generalized sort of data model that can suck in all of that data.
Cool.
Does that kind of answer your question?
It does, but if you choose Epic, then you'll be designing an integrator for that Epic instance.
Right.
And then you might need different ones for different.
Yeah, exactly. Yeah, and we got to start somewhere, right? So we started in Germany with the electronic medical records there, and so we're going to find something.
It's probably a good system to start with.
Probably better than Epic.
Simpler system. Any other questions? Don't start in the US.
So you guys have already been working on type 2 diabetes. What's next on Stanford's radar? What's the next kind of big
So the type 2 diabetes, it wasn't an intentional choice to choose type 2 diabetes. They gave us a list of 8 diseases, and it just so happened that the genetic disparity across populations for type 2 diabetes was so apparent that that's the one that they kind of focused on. Our next step for that is to actually take the newest 1,000 genomes data set, which is the 2,500 individuals, and do that same test again to see if do we see that same disparity with more individuals. And we'll probably do that with all diseases in variant because, again, it doesn't take that long to do that.
If there are no more questions, then we'll move on. We'll say thank you.